A startup from Berkeley, California, Sugarlogix makes human milk oligosaccharides, which are found in breast milk, via microbial fermentation.
So me and my co-founders, we met through a biofuels project funded by British Petroleum, and I was pursuing my PhD in chemical engineering at UC Berkeley, and I was working on the process development side of producing biofuels, and the rest of my co-founders were working on the biology side to create microorganisms that could produce biofuels efficiently. And I was about to graduate. We wanted to start a company, and by that time the biofuels industry was fast declining, so we decided to pursue another product, and one of our co-founders had a side project making HMOs out of E. coli processes. So
Okay.
that's why we decided to adopt that process and then to incorporate with our yeast-based fermentation production process.
So there are lots of different human milk oligosaccharides. Which ones are you looking to produce and why? Do they all do different things?
So there are over 180 different types of human milk oligosaccharides, and the concentrations is very unique to each mother, and there still needs to be a lot of work and study to be done to fully understand the depth of what they do in humans. So the most widely available HMOs have been studied the most. So we will target the HMOs that have been studied the most, and start with the simple and the ones with the highest concentrations, and then moving on to the ones that have been less recognized. So the most obvious use case for the HMOs is in infant formula. Obviously, nature is trying to recommend it by putting it inside human breast milk. Additionally, we're trying to research additional usages of the HMOs. Adults in the current world, they're faced with a lot of bad choices that is not recommended by Mother Nature. For example, we smoke, we drink alcohol, we get stressed a lot. So adults in the current world could use every little boost that we can. So we're trying to do research in adult applications as well.
Can you explain why HMOs are of interest? Is it because they stimulate the growth of particular kinds of bacteria that we know are very beneficial to the infant gut, for example?
Yes. The HMOs are not meant to be used as energy sources for the human body. Rather, it feeds the beneficial gut bacteria very selectively inside the human gut. So what it does is it sort of starves the bad bacteria and then promotes the growth of the beneficial gut bacteria, which in turn helps our gut flourish, and which is also connected with our nervous system and our immune system as well. There are quite a few players in this field, and it's not actually just the HMOs. There are a lot of missing components that are very unique to human breast milk that is just very difficult to produce outside of a human body. So many different players are approaching using different approaches to address the different types of the problems, and with HMOs as well, a few players are adopting E. coli based production processes to manufacture HMOs. A few players like us were using the yeast based system to produce making the HMOs. Some players try to cut up larger chains of sugar molecules and then to break them into smaller chains of human milk oligosaccharides. So lots of different approaches.
Right. Okay, so as I understand it, your process, I assume, would use a genetically engineered yeast that would then, you're kind of giving it instructions to produce these sort of target substances. Is there some pushback against companies producing, food ingredients that involve genetic engineering, even if, the yeast isn't present in the final product?
Mm-hmm. So two aspects we use to, communicate why our organisms or our products are safe and thus should be used, can be used widely in infant formula. One is that it contains no genetic material whatsoever in the final product. Mm-hmm. So there is no difference between the molecule that we make and the molecule that is found in human milk. So that makes it very safe. Additionally, we're not just introducing the genetic manipulations to, I don't know, for example, cut cost for us. We're trying to do something that is just very difficult to do outside of a human body, thus the need for genetic manipulation. So I think that really justifies our case to have to adopt to this means of processing.
Great. So give me a sense of the stage of development you're at in your business now. Are you still in the R&D stage? Do you have a product? When might there be a commercial product available?
So we're somewhere in between. We're currently trying to scale up our lab scale R&D process into larger scale sized facilities so we can manufacture more and more. Start with samples, of course, and then moving on to producing products that can actually be used for human usage. Right now, we don't manufacture at food grade facilities, so all of our samples, they're not meant for human use, but more in vitro studies and animal studies. But in the future, we aim to start producing in GMP grade facilities.